Hi, everyone, and welcome to what we are reading as part of our YouTube channel of the South America Mycorrhizal Network. And today we have Dr. Katie Field from the Plants, Photosynthesis and Soil School of Bioscience at the University of Sheffield. And today we're going to be discussing your recent paper, direct nitrogen, phosphorus, and carbon exchanges between Mucoromycotina, so the fine root endophytes, fungi, and the flowering plant in novel monoxenic cultures. I've been looking at your work for a while. It's so interesting, and you've been working with liverworts, and then you've been working also with these fine root endophytes, the Mucoromycotina. Can you tell us what led you to work with these organisms first?
Sure, yeah, thanks, Pedro. Really nice introduction. It was entirely almost by accident, I guess, really. So I first started working with arbuscular mycorrhizal fungi, looking at sort of functionality and changing CO2 concentrations and how a high CO2 atmosphere in the Paleozoic might have affected the functionality of early mycorrhizal associations in like the first land plants, using liverworts as like a model system almost, I guess, to sort of hypothesize about how that might have affected it. And we were firmly focusing on arbuscular mycorrhizal fungi. And then there's a group of liverworts called the Haplometriopsida, which they came into focus sort of, I think it was in 2011, so it's a long time ago now. But Martin Bidar-Tondo, he and his colleagues, they actually they sequenced the fungus that was growing and living inside these sort of very early divergent lineages of liverworts. And it turned out they weren't arbuscular mycorrhizas at all, but they were actually Mucoromycotina fungi. So then, I mean, I've worked with Martin for a long time, and in discussions and stuff, the question came like, well, what are these fungi doing? If they're not mycorrhizal fungi, why are they living with them? Why aren't they causing disease, etc.? So we were like, okay, let's do some experiments. And so we did some experiments with these fungi in liverworts, and we found that they were mutualistic. There was carbon and nitrogen, carbon, phosphorus, and nitrogen exchanges between them. And that really was the start of the whole line of research. So then there was a publication in 2017 by Suzanne Orchard, where she actually made the first link between these Mucoromycotina group of fungi and the fine root endophytes. And it was the same Mucoromycotina fungus in the plants that she was looking at as it was in our liverworts. And so that's when the light bulb went off in our heads, and we were like, oh my goodness, fine root endophytes, they're Mucoromycotina fungi, and they're functional, and they're doing this stuff in liverworts. But then I think it took an awful long time then to actually isolate the fungus and culture it, and then be able to use it in sort of monoxenic systems and really unequivocally prove that it was this nutritional mutualist, which is what our latest paper shows. Like, it's really exciting.
Yeah, so these fungi, you isolated them first from liverworts. In this particular paper, you isolated them from a moss, right? And from Lycopodium.
Yeah, so it's a lycophyte, so it's a vascular plant.
Mm-hmm.
Yeah.
And you isolated these fungi, and you were able to identify it and then introduce it into or inoculate it into clover in a novel in vitro system. But even before we get to talk more about this paper, one of the things that really interests me. So before these fungi, we see them all the time or very often when we stain roots, we used to call it Glomus tanu, right?
Yeah, yes,
so it turns out that it's these
totally
fungi
different thing.
Yeah. So how widespread are these fungi? Do you think?
Great question, Pedro. We don't know. I mean, if we consider that everything that we've previously looked at down a microscope and thought of as being fine root endophyte, ergo Glomus tenue, then I mean everywhere, right? The state of the art at the present day is that we're finding it in almost all plants that host AM. They also host fine root endophyte, or these Mucoromycotina fine root endophytes is what we're trying to establish that we call them, so that it avoids confusion with our arbuscular mycorrhizas. So we've got these 2 distinct groups that potentially have discrete different functions. But yeah, they seem to be everywhere. The habitats are very broad. They seem to like disturbance. We seem to get a higher abundance in wet environments. So I'm currently leading an ERC-funded project to investigate this exact question, to look at the distribution and the host range of these fungi. So we're actually conducting this sort of Europe-wide sampling campaign where we're looking for, we're sequencing the fungi in the roots of a whole range of different plants that are common across different habitats, but also some that are specialist to different habitats as well, to try and get an understanding of the host range, the environmental preferences, the ecological niches, but also the connectedness of them. So doing some network analyses to try and look and see like how related are these different endophytes, and how do they interact with each other? Right. So yeah, answer if you don't.
Yeah, they're probably
we think everywhere.
Yeah, everywhere. So I think that answers the question. And in your study, so yes, for the first time, I guess you were able to demonstrate, okay, we can work with these fungi in extended cultures and see directly how they contribute to the plant. I love how elegant your experiments are done, and how well designed they are. I mean, you isolate a fungi from it's true. You isolate a fungi from a vascular plant from Lycopodiella, and then you introduce this fungi to clover. You use molecular tools to identify it. You're able to get these petri dishes and grow the clover, and then have the plates with this fungus colonizing the plant in pure culture, and then you detach portions of the phytogel and you add N15 or phosphorus 33, and then at the same time, I think you also add you isolate the bottom of the plate, and then the top where the shoot is exposed to the atmosphere, you add CO2 14. So to really look at the exchanges between the contribution of these fungi to the plant, and then the contribution of the plant to the fungi. And in your paper, if I remember correctly, you supplied the medium with phosphorus and nitrogen, and you saw that just within 24 hours, that like almost 50% of what you supplied to the plate was transferred through the fungi to the shoots of the clover. So could you comment on that? And not just the phosphorus, but also the nitrogen really interests me as well, because these fungi seem to be facultative saprotrophs, right? So and I know that in your paper, the concentration of nitrogen there was a trend for nitrogen, although uptake is really so. Anyway, did the plants really grow a lot more with these fungi in terms of biomass? And then what do you think are the implications in terms of nitrogen, phosphorus, and also the costs in terms of carbon?
My goodness, Pedro, so many questions in one.
A lot here.
Need to break that down a bit, I think. So the first thing I want to point out, like very clearly, is that obviously this isn't all my work, right? So I work with some really talented collaborators. So for this particular piece of work, and this paper in particular, it was such an amazing collaborative effort. So Sylvia Pressel at the Natural History Museum in London, she isolated the fungus from Lycopodiella and cultured it and came up with that first breakthrough. Martin Birartondo developed the primers and was able to then sequence it and then identify and relate it to what we'd done previously. So my role in this was really to design the experimental systems and do the isotope tracing and the labeling of those systems. So it was a proper collaborative effort, and working like that is so joyful, isn't it? Like you each have a distinct expertise and contribute, and it's way more than anything you could do on your own. So yes, so that's kind of what each of us did in that particular experiment. In terms of what we found, yeah, it was really surprising, but I think it's also really important that we don't, overinterpret what we found from these systems. We need to remember, particularly when we're working in things like Petri dishes and with no soil, there's no other microbes. It's really, I feel like these experiments are really about they're more of a can it rather than a does it type question, if that makes sense. So we're testing whether it can do something or not.
For sure. Yeah.
Now, I struggle then to make a really strong link to what it actually does do in nature, and when other plants are present, when there's other fungi present, and there's changing environments, and there's different levels of soil nutrients, etc. Because in these systems, we had a fairly low nutrient agar-based medium. I'm sorry, it was a phytogel-based medium, not agar, beg your pardon. And then we introduced a set amount of phosphorus and nitrogen within discrete little wells within the media, and then we just measured how much of it moved and went to where it went. I was really surprised how quickly it moved in those systems. But I mean, yeah, I was glad it did move that quickly, because the conditions that we were doing these experiments under, it was the UK lockdown had just been announced for COVID. And so I had to scramble basically and make those systems work really quickly. I had to go to the Natural History Museum and rescue them from the museum before it closed to everybody for months, and we would have lost them. And then I had to get them back to university and do the experiment instantly so that they would be radioactive, because then our health and safety team meant I would have to be allowed to go in and finish the experiment because it was radioactive, right?
Oh yeah, I saw you had a Geiger counter.
Yeah, so I was like, right, I'm just going to do it right now.
Yeah.
So thank goodness it did work really quickly over the course of a weekend. And then I went in on Monday and they were radioactive, so I was able to then do that carbon trace. But I guess the reason it's that quick in those systems is just that there's no barriers to it. There's no competition. The plant needs the nutrients. It's in this low nutrient medium. And the fungus isn't really having to work very hard to get it. There's no competition. Yeah, and I was surprised, I think, having seen the huge amount of phosphorus that was transferred. I was surprised when the data came back from the mass spectrometer that the nitrogen wasn't as clear cut. And part of me wonders if that's to do with the plant itself being a legume, and perhaps it doesn't rely on fungal nitrogen transfer as much as it would do as another non-leguminous plant that can't fix nitrogen from the atmosphere. Now, we know the plants in our systems weren't fixing nitrogen from the atmosphere. They had no nodules. They were exenic systems. But maybe the plant just is able to assimilate more of it on its own. Data we've got from sort of soil-based systems and experiments I've been working on since using these cultures and different types of plant suggest that nitrogen is a much more kind of important nutrient for these binary endophytes than the clover experiment suggests. So
nice, yeah.
We preview into some of the stuff that we're going to be coming out at some point soon, hopefully.
Yeah, that brings me to the next question. I guess being facultative saprotrophs and coexisting with arbuscular mycorrhizal fungi, from your work, I guess it's plausible to hypothesize that maybe these fungi don't really compete with the Mucoromycotina, and maybe they are complementary. Is that a...
Yeah, I mean, I guess there's a bit of both, isn't there? Because I think in terms of function, they're complementary. There's definitely complementarity between them. So we see when we have dual colonized plants that the plant really does benefit from the best of both worlds. But I mean, there is an element of competition inherent because they're both endosymbionts. They're both trying to occupy the same space within a plant root, right? So they form structures within cells. We haven't seen any evidence that they occupy exactly the same cell at the same time. So whether they're, I don't know, is it a case of one gets there first and occupies it, and then the other one goes somewhere else and occupies something else? Or is the plant somehow in control of where things are colonizing and which ones are colonizing where? I don't know. And does that change when the environmental conditions change to suit it best?
Yeah, your
There's
cytological
so many
work.
questions.
Yeah, exactly. Like your cytological work, I think in your 2012 paper, no, 2015 in New Phytologist, demonstrates that for one of these fungi that you worked with,
Mm.
and there were changes in the morphology of the plant. So they actually
Yeah, that was
alter
the
plant
liverwort.
traits.
Yeah.
Arbuscular mycorrhizal fungi don't really do that. So
No.
how can you comment a little bit on the on the morphology? You also they form these arbuscule-like structures. You call
Yeah.
them
Okay.
lobe structures. So could
Yeah.
yeah.
So yeah, a bit of back. So that paper where that was the first one where we were working with these Mucoromycotina fungi, and we had not made the link between them and the fine root endophytes yet. So we thought this was, well, we're working to a hypothesis that this was a weird fungus that colonizes weird sort of plants, and it's not very frequent. It just seems to be these liverworts at the sort of base of the phylogeny. Yeah, so the terminology, we were really careful in that paper to be using very very specific terminology to describe what it looked like without trying to overlap it with the arbuscular terminology. So when we say arbuscular-like structures, because it's not an arbuscular mycorrhizal fungus of the Algoma, we're not calling them arbusculars, we're calling them arbuscular-like. Now, I mean, maybe that changes, I don't know. So yeah, our terminology is quite deliberate and quite distinct from how you might describe sort of like a glomeromycete. Yeah, and I mean it changes all the time. So the latest stuff we've been looking at. So one of the things that we had great fun was trying to describe what the vesicle-like, but they're not vesicle structures. And so I think we settled on lumps. We called them
Yeah, or
lumps.
infaradical spores. Yeah, yeah.
And they were like degenerating lumps and all sorts of wonderful things. But now we're not sure, we're even more unsure what they are, whether they're actually spores. They seem to have lots of nuclei in, or are they fat sort of storage organs akin to vesicles? So they also have a lot of lipid inside them. So we haven't seen any of them germinate in culture. We can produce a lot of them, but we don't see them germinating. So I mean, there's still so much we don't know about this fungus. And I
Right.
think yeah.
And the one you isolated, you did molecular identification, and it came out as endogonal SP, which is different than the fungus that used to be named Glomus tenu. So how I guess it was the one that we used to be Glomus tenu is Platyconsortia tenu. It was renamed, but this one that you worked with in this paper is not that one. Can you comment on the diversity of these fungi and how similar they are to?
Absolutely, and it's a work in progress. We don't know. We know that they are diverse. We've got evidence that they're diverse, right? The ones that we're sampling in the field are different to some of the ones that we got from New Zealand, which are different from the ones we sample in Yorkshire. But I mean, yeah, I wouldn't like to comment too much on how diverse they are because we don't really know. We haven't sequenced. We're in the process of sequencing the genome of that isolate at the moment that we used in the New Phytologist paper that just came out. But yeah, until we have more isolates, either from the same plants or from different plants, it's really difficult to know. I think maybe that's a question to come back to in maybe 3 or 5 years' time.
That's awesome. So, we talked about the difference between phosphorus and nitrogen, facultative saprotrophs, in terms of plant biomass. So these are clearly mutualists. They talked about competition. I had other questions here. I guess we talked about the morphology, the changes in morphology, which brings me to traits. How different, what's the life history based on what your observations? Do the hyphae, the extra radical hyphae of these fungi go really far out? Are they a lot, or we don't know any of these questions yet?
That's why we know nothing. No. So in culture, I can talk about the stuff that we've done in culture. So they do seem to change the way that they grow depending on what resources are available.
Yeah.
So they seem to have like a foraging phenotype if there's a low nutrient status. If you put them in a very rich medium, they get really fat and dense. They produce a lot of these maybe spore-like structures. I'm going to call them what used to be known as lumps.
I see. Yeah,
And then, yeah, I mean, we don't know. I mean, the trouble is in the soil, they're so fine. They're even finer than AM hyphae, right? So I don't know if we'll ever know how far they extend. It'd be a cool experiment in culture. You can grow them maybe 5, 6, 7 centimeters away from a plant root. But again, what can you really say about a natural system from a culture dish? You can say if it can or not, but maybe not so much whether it's how relevant it is.
It's really, it's really hard. It's early days, I guess.
Yeah, it's exciting though, right? I mean, it's lovely. It's amazing to have a system where you know so little, you know it's important, you've shown that it's important, but you know, it makes you rethink everything that we know about AMs, having, you know, maybe we've confused some AM stuff with binary endophyte stuff, and actually some of the stuff we've attributed to them is actually this whole other thing. Yeah, and
when we have collections of in pure culture of mycorrhizal fungi, Algoma Mycota, and then we know the plants are maybe the symbiosis is a lot more than that. It includes these other taxa that are not in collection. I guess maybe we will also need to have collections of these Mucoromycotina fungi, I guess.
Maybe we have the first one, yeah. We've got the founding member in Sheffield now.
So I have another question. These again, like you were able to isolate these fungi from plants that live in northern, colder environments and put them in an agricultural plant, clover, I guess. So, you know, they're obviously not host specific, or maybe they are to some extent.
Isn't. Yeah, I mean, this one isn't. We know some plants can be specific. So there are, from what we've learned so far from all of Martin's work, and also some of the work that we've been doing as part of this ERC project, is there are some plants that seem to form associations with M-Fray, I'm going to call them because I can't be bothered to keep saying Mucoromycotina, that form associations with the M-Fray that don't with the AMF. So with the liverworts, for example, so the Haplometrium and the Troibia, they only ever associate with M-Fray. Like Podiella and Undata, that never has an AM. We've never detected AM in it. It seems to only have M-Fray. Yeah, so it'd be really interesting to kind of start looking at plants that are classically what we think of as being non-host plants and see, you know, can they form associations with this fungus?
Especially being a saprotroph.
Yeah, I think what's really exciting then is kind of the evolutionary inferences from that then, isn't it? It's kind of, especially with some of these liverworts and sort of, I don't want to say primitive, but like the systems that we use to compare what early plants would do. So some liverworts or early vascular plants or something. You know, is it doing things in a different way to AMF? I mean, we've done a lot on AM signaling and AM genetic toolkits and stuff, but maybe they're doing something differently, or they're doing more of a like a pathogenic type style of colonization.
And that there's what I mean, if we consider that the evolution of these fungi occurred 500 million years ago, early Devonian, even before then at a time when CO2 was really high, but also, I mean, we don't know for sure, it's all proxy data modeling, but temperatures were average temperatures. Today they are global temperatures 14 around there. Back then, so CO2 over 1,500 ppm, temperature 20 degrees, 25 degrees average, so a lot hotter, a lot more CO2. And yes, these fungi, I guess, they are facultative saprotrophs, so they were already on land. So these could have been perhaps the first fungi that were helping the first early plants. It's as a hypothesis, and
Maybe, or
then
a common ancestor of them, maybe as well.
Mhm.
So the classic view is that it's sort of AMs diverged around the same time as land plants, and they colonized the earth, and AMs helped them get onto the land, la la la. But you know, the phylogenetic placement of these fungi, together with their patterns of colonization in plants today, and being so widespread and everything, you know, they also have to be candidates for being important in sort of floral history of Earth, right? I mean, or at least they're common ancestors. Maybe there was a common M-Fray AMF ancestor that was actually the first symbiont, and then you have the divergence of the obligate versus facultative biotrophs. I don't know. It's certainly a really interesting question. It's kind of, I find it really, I don't know, I enjoy thinking about it and kind of coming up with all the different possibilities in your head, and then kind of it
I was
blows your
thinking
mind a
more
bit.
of I was thinking, yeah, sorry to interrupt.
No, go ahead.
Yeah, I was thinking more of in the sense of because these fungi, the implications of your work for in terms of ecology and also climate change biology, the phenotypic plasticity of these fungi, knowing that maybe they evolved at a and with plants that could cope with much warmer conditions, a lot of CO2. Do you think that's
Yeah, no, absolutely. I think, yeah, I mean, it's kind of an ongoing theme in all the research I've done, is to kind of push the extremes of what these fungi do under different climates, right? So with the AMs, I've tested those under like high CO2 and then modern CO2. So I think, yeah, we need to do more research on these M-Fray and see, do they have a similar plasticity? We did some work with the lycophytes. So Grace Hoysted, was a postdoc with me a while ago, she's now at UCD in Dublin. She did some work with Lycopode yellow inundator, and she looked at it under a high CO2 atmosphere and did the carbon for nutrient exchange experiments in soil-based systems. And I mean, she found it didn't change what it did. It maintained nutrient transfer, and it didn't take more carbon. So, I mean, it's really hard. I mean, maybe it's different in different plant species. It's a really complex picture, and you can kind of only really start understanding that when you really you do the experiments and you do the laborious sort of tracing experiments and stuff, and actually test the boundaries of what's possible, I guess.
Yeah, awesome. Maybe one last question. I'm very curious about what your thoughts are on this. So, we talked a little bit about consequences in terms of fundamental ecology. How about the consequences? Now you've demonstrated for the first time that these fungi, in absence of bacteria or other organisms, they actually mutualists. They have the potential to transfer phosphorus and nitrogen. So then in a world where there's so much interest in inoculants and applications, what are the consequences of these findings for in terms of applications?
Yeah, well, I mean, potentially massive, right? I think, yeah, I mean, we need to know more about their ecology and kind of, you know, what environmental conditions and constraints do they operate under. And again, the host range question is really relevant here. And also the diversity question is really important. I think we need to consider also the whole mutualism, parasitism continuum, and think about, you know, do all M-Fray fungi fall on the mutualistic end of that, or actually some species more parasitic on some species of plant than others? And I guess it's all of the same considerations that should be borne for application of AM fungi in sort of commercial situations as with M-Fray, right? That's not saying they necessarily are.
Exactly.
But yeah, I guess it's all of those same considerations. So you also have that whole little sort of line of argument that actually, should you be introducing foreign fungi into you know, in potentially ancient soils and environments, and what happens if they escape? Do they run rampant? Is there any, what are the unintended consequences of that? In reality, I imagine it's hard to assess, isn't it? I mean, we've gone through decades and decades and decades of AM research, which also encompasses Mucoromycotina,
Yeah,
and
exactly.
we can't distinguish it. So I think, I mean, for me, one of the major, it's both, it's simultaneously exciting and intimidating, I think, is when you start looking at previous experiments where functionality experiments, basically, where you've got different experimental systems which have used either a known inoculant, which is known species, it's from culture and it's been sequenced and it's bona fide AM, soil-based culture where you don't actually know exactly what it is and you haven't sequenced it in some of the older studies maybe, and functionality and structure and ecology has been inferred from these and been assigned to AM only. And I think one of the major challenges now is to untangle what is AM, what was M-Fray, how are they distinct, how do they interact? Yeah, there's a really interest. It's yeah, like I said, simultaneously exciting, but also quite intimidating because there is a lot of research to go through and pick
It's
out, like, who used what inoculum and what was done with it and what were the conclusions and stuff. So yeah.
exactly. And yeah, there's a lot. Yeah, your work opens, it's one of those papers that opens a new field, a new air, like a new
Yeah,
branch, I guess, in symbiosis
thank you.
to
It's really
explore.
exciting.
And I love the pioneers, the pioneering work that yeah, that
Yeah.
advanced the field like that. So
Oh, thank
yeah,
you.
thank you so much. I mean, yeah, do you have any other comments?
Um, no, I don't. I think it's, yeah. I would be really, I don't know. I'd be really grateful and excited to the community if people started sort of including M-Fray in their analyses and started thinking about it as another mycorrhizal fungus. Um, and there's so much to be done, and there's so many different skills and hypotheses and sort of opinions that need to be explored in relation to this now. And like, kind of, it's almost like, yeah, we really need the community to kind of pick this up and run with it now and like really explore what's possible. And I mean, I think the point you made about applications is really relevant because we talk a lot about enhancing mycorrhization in farming, for example, and in reducing what's the word, synthetic application of synthetics, or enhancing sustainability, I think is what I'm trying to say.
Mm-hmm.
And maybe M-Fray could be a key player in that.
Yeah, and
But
it apparently,
until we understand its ecology and biology, then it's an untapped resource, isn't it?
yeah, and apparently they can access nitrogen from organic sources, right?
Yeah, and this is something AMs can't do. So I've got a PhD student at the moment, Alex Watts, who's working on his project is investigating sort of the saprotrophic capabilities. So he's testing to see what exactly can this fungus do, like what sources of nutrients can it get, how is it doing it, and then what are the consequences for its plant host in those situations. So
That's awesome. I find that
yeah,
fascinating. And the traits, maybe these tiny little hyphae being able to
mm,
explore, to exploit areas in the soil that maybe AM can't even access. And if they can
yeah,
access organic forms, it's like I said, it opens,
it really
and
does.
it's
Yeah.
you're absolutely right. I think the community needs to have M-Fray. I like how you call them. I was
Oh,
always
thank you. That
find
was
root
Sylvia
endophytes.
Pressel's
M-Fray is just easy. You can
Emperor.
maybe
Yeah,
just
that's
adopt
Sylvia
that.
Pressel's mentioned that one. Yeah, yeah, no, I like it. I think it's also like, I mean, this is one of my one of the things I always find I have to sort of address in reviews for papers and stuff is like what we call them. And I think it is really important that we distinguish them from AMF. Um, I'm very, very, very reluctant to kind of take up sort of AM terminology to describe this entirely distinct fungus.
Mhm.
Um, particularly because in the past, like, there's so much muddiness in the literature about what is a fine root endophyte, what's an arbuscular mycorrhizal, and then they all change names and they all change positions all the time. I think at least if we can distinguish when we're talking about Mucoromycotina, we're talking about Mucoromycotina fine root endophytes, M-Fray. And when we're talking about AMF, we're talking about Glomeromycotina, AMF. Mm-hmm. I think going forwards, I think making that distinction is going to only help sort of clarify all of this stuff that we're having to try and untangle.
That's really important. That's a really good point.
Yeah.
I think this really covers the range, and I just noticed also that I didn't log in with my other account, so this one ends actually in 3 minutes. So
that's good timing then.
That's perfect timing. Cesar also told me that this should be kept to 20 minutes, but the topic is just so interesting, and we could be here talking all day about
well, hopefully one day, Pedro, we can, hopefully.
Yeah, for sure, I'd love that. But and you are organizing the ICOM. I definitely want to go. Yeah.
Good. Well, we shall be in touch about that soon, I'm sure. But I'm really hoping as part of that we can put in like an alternative mycorrhiza session, and kind of really, because I think in a year's time there's going to be so much more stuff that we know about this.
That would be awesome, actually.
Amazing.
Cut me in. Yes.
Yeah. Excellent. Good to know. I'll pass it on to Dave Johnson. Lovely. Excellent. Okay.
Thanks so much.
No problem. Thank you. It was really nice to chat. And hopefully I'll meet you in person one day.
Yes, likewise. And in the meantime, I'll keep reading your awesome papers.
Thank you so much. That's so kind. Take care.
Thank you. Bye-bye.
Take care, Pedro. Bye.